Okay, let's dive right into this explainer. Today, we're exploring the absolutely fascinating history and techniques behind microbiology, straight from our study notes. We're talking about a world entirely invisible to the naked eye, yet one that literally runs everything around us.
I mean, think about it for a second. How do you identify an enemy you can't even see? It's kind of like trying to fight a war when you have no idea what your opponent looks like or where they're hiding. That was the ultimate puzzle for early medical pioneers. Devastating diseases were spreading, people were getting sick, and honestly, nobody knew why. Today we're going to see exactly how they built the solutions from scratch, literally inventing the tools to see the unseen.
Here's our agenda for today. 1. Pioneers of microbiology. 2. Modern microbiological breakthroughs. 3. The principles of staining. 4. Decoding the gram stain. And 5. Special stains for tricky microbes.
All right, let's kick things off. Section 1. Pioneers of Microbiology. It is just amazing to trace this timeline, you know? It shows the rapid evolution of human discovery. We kick off way back in the 1590s with the Janssen duo. They combined lenses to create the first compound microscope. Then, jump to the 1670s. Anton von Leeuwenhoek uses a simple microscope and becomes the very first person to actually see what he playfully called animalcules. Incredible, right? Fast forward a bit to 1796 with Edward Jenner's smallpox vaccines, culminating in the late 1800s with Louis Pasteur and Robert Koch. These two established modern germ theory, finally proving that these tiny organisms were actually causing disease.
Now, to easily recall Louis Pasteur's massive contributions, just remember this fun phrase from the source notes: Father, car vaccines. It's pretty catchy. He is the father of microbiology, and he developed vaccines for cholera, anthrax, and rabies. CAR. It's just a wonderfully efficient way to lock in a cornerstone of medical history.
Now, while Pasteur was making vaccines, Robert Koch gave us Koch's postulates. These were the standard, highly rigorous rules for proving that a specific microbe causes a specific disease. But biology is messy, right? Rules get broken. You absolutely must know the rule breakers. And the notes give us another great mnemonic for this: LPG. That stands for Mycobacterium lepra, Treponema pallidum, and Gonococci. These exceptions just won't grow in standard lab cultures or follow Koch's strict rules. It proves that microbes are literally always challenging us to adapt.
Moving right along. Section 2. Modern microbiological breakthroughs.
These modern legends give us incredible breakthroughs that really form the backbone of modern medicine. First up, Alexander Fleming, who gave us penicillin, ushering in the antibiotic age. Then there's Barbara McClintock. She totally revolutionized genetics by discovering transposons, or, as they're commonly called, jumping genes. Next, Carrie B. Mullis gave us PCR, the polymerase chain reaction. That is the absolute gold standard for replicating and testing DNA today. And of course, we have Houghton, Alter, and Rice. Their Nobel-winning work didn't just find the hepatitis C virus, they actually mapped out exactly how its mechanism works in the human body. So with the who established, it's time to shift gears to the mechanical, step-by-step how of identifying these microscopic organisms.
Section 3. The principles of staining.
Looking at the hand-drawn diagrams from the source material for the gram stain and Ziehl-Neelsen procedures, it really reminds us that stains aren't just about painting cells with pretty colors. Not at all. They are highly targeted chemical interrogations of a bacteria's biological defenses. We're using chemicals to reveal their hidden structures. But before you can interrogate a microbe, you gotta lock it in place on your glass slide. We call this fixation. You can use heat fixation, which is literally passing the slide over a flame to stick the cells to the glass, or you can use chemical fixation with methanol or alcohol. Once they're locked down tight, you can apply a simple stain like methylene blue just to wash the slide and see the basic shape and arrangement of the cells.
All right, Section 4, Decoding the gram stain.
This procedural life hack relies on a strict 4-step sequence. First up, apply the primary stain, crystal violet, for 1 minute. Bam, everything turns purple. Second, you add the mordant, grams iodine. This acts as a trap to lock that purple dye inside the cells. Third, and pay attention, because this is the absolute most crucial step, you wash the slide with an alcohol or acetone decolorizer for just a few seconds. Finally, step 4, you counterstain with safranin for 1 minute.
So the real magic happens during that alcohol wash, and it all comes down to the cell wall. Gram-positive bacteria have thick peptidoglycan walls that act like a giant fortress, trapping the purple crystal violet dye. But gram-negative bacteria, their walls have a high lipid or fat content. The alcohol actually dissolves those lipids, essentially blowing a hole in the wall and washing all that purple right out. When you add the pinkish-red safranin counterstain, only the newly colorless gram-negative bacteria soak it up. Instantly, you've categorized them. Purple is positive, pinkish-red is negative.
But hold on, remember, this amazing toolkit has blind spots. You really can't rely on it for everything. Certain organisms simply won't take the gram stain properly. Take mycoplasma, for instance. It's the absolute smallest bacteria and it lacks a proper cell wall entirely, so there's literally nothing to trap the dye. Then you have rickettsia and chlamydia, which hide deep inside host cells, making them super hard to reach. In spirochetes, they are just too remarkably thin and slender to hold enough dye to even become visible.
Which brings us to Section 5, Special Stains for Tricky Microbes. So what happens when the gram stain fails? Well, for heavily armored bacteria with thick, waxy lipid barriers, like the one that causes tuberculosis, we use the Ziehl-Neelsen, or acid-fast stain. We literally use heat to melt the barrier and force the red carbofuchsin dye inside. Now, I absolutely love this mnemonic from the notes to memorize the acid-fast organisms it targets. My nose is cold hot. It brilliantly triggers the recall for mycobacteria, nocardia, isospora, cryptosporidium, and hydatids cyst hooklets. Good luck for getting that one.
Alternatively, sometimes it's better not to stain the microbe at all. Enter the negative stain. This uses India ink or nigrosine to stain the background dark, which perfectly silhouettes the unstained protective capsule of a fungus. Or, for those ultra-slender spirochetes we mentioned earlier, we use impregnation stains, like silver stains. The silver chemically deposits directly onto the surface of the bacteria, physically making it thick enough to finally see under the microscope.
This table perfectly organizes the rest of our specialized palette, showing exactly how we match the chemical to the biology. We use the Albert stain specifically for diphtheria, which turns the bacilli green with distinct purple granules. We use Flagella stains with basic Fudgeson dye to color a bacteria's tiny movement tails bright red. And the Spore stain uses heat to force malachite green into highly resilient, dormant bacterial spores, leaving the rest of the vulnerable organism colored red with safranin. It is an incredibly precise chemical process.
As we wrap up this explainer, I really want to leave you with a provocative thought. It is just mind-blowing to consider how far we've come, isn't it? From Anton von Leeuwenhoek marveling at tiny animalcules through a crude glass bead, to modern pioneers literally sniping and editing the genetic code of life itself with CRISPR-Cas9. None of it would be possible without the pioneers who figured out how to make the invisible visible. Keep that curiosity alive and keep digging into the details.